Hello students, welcome back to our channel with another video. This is Dr. Isha Bey and today we are going to be solving Cambridge exam IGCSE Biology 0610 paper 4-2. Alright, so let's begin, but first let me introduce you with the rules and regulation of this paper. So this paper is known as paper 4, that is actually the theory based paper, and attempt all of the questions in this paper. Alright, so let's begin with solving this paper. So question number 1 says that Figure 1.1 is a simplified diagram for circulatory system in humans. Part 1 says, state the letter from figure 1.1 that represents a blood vessel that contains blood vessel that contains the blood with the highest concentration of the oxygen. So, obviously, anything that will be coming from the lungs will be having highest concentration of oxygen, yes or no? So, what is a blood vessel that is coming from the lungs to the heart? That is pulmonary vein, and that is B. Alright. So the answer to this question would be B. Now, part 2 says state the letter from figure 1.1 that represents hepatic portal vein. So hepatic portal vein is basically the blood vessel that carries the blood from the gut, this is the gut, to the liver. Alright, from the gut to the liver. Alright, so what is a blood vessel that is carrying the blood from the intestines or the gut to the liver? That is C, and we name it as hepatic portal vein. Alright, I hope this was clear. Pardon my writing. Alright, so part 3 says state all the letters from figure 1.1 that represents the artery. So here you need to focus on the question, what is actually question demanding. Question is asking you to jot down or to separate all of the arteries and to write down the letters representing arteries over here in this space. Alright. So first of all, you should know that arteries are the blood vessels that carry the blood away from the heart. Where? Away from the heart. Alright, so we need to figure out all of the blood vessels that is carrying the blood away from the heart. So obviously B and C will gonna be omitted. We are not going to focus on B and C and waste our time. We are not going to do that because we know that they both are the veins. Alright. So let's see what else is going away from the heart. Is this an artery? Can we call it the artery because it is going away from the heart? Yes, as we can see that the arrow is away at D. So the arteries, the vessels carrying the blood away from the heart are A, D, and we can also focus over here that this is also the blood vessel that is carrying blood away from the heart, that is E. So A, D, and E. So this is the answer, that vessels A, D, and E represents the arteries. Now let's move on to the part 4. Explain the importance of the part labeled Y in figure 1.1. Alright, so what is Y? Part labeled Y. So Y is this. And what is it actually labeling? It is labeling this. That is the septum. That is the interventricular septum. So septum is basically actually separating the left side of the heart from the right side of the heart. So this is preventing the mixing of the deoxygenated blood with the oxygenated blood. And it separates the atria from the ventricles. It separates the left atrium with the right atrium, obviously, which means it separates the left chamber from the right chamber. So it keeps the blood separated and it helps to maintain the blood pressure. Alright. So what are we going to write down in the answer? This is what we are going to write down in the answer, that the septum separates the left side of the heart from the right side of the heart. This will give us 1 mark, and the second mark for telling the function, another function, that it keeps the deoxygenated blood away from the oxygenated blood. Alright. Second mark for telling that it keeps the oxygenated blood away from the deoxygenated blood. Alright. So we will be going. Let's move on to the question number 5 that says that state the evidence from figure 1.1 that the diagram shows a double circulatory system. So we have to tell that how does this diagram shows that this circulation is a double circulation system. So what actually is double circulation system means? Double circulation system means, wait, let me change the color of my drawing. So the double circulation system means that there are two circuits. There are two circuits in one complete cycle of the heart. Alright? In one cycle, the blood from the heart is pumped towards the lungs and from the lungs back to the heart. This is the one cycle. And then in another cycle, after coming to the back to the heart, the blood is then pumped from the heart to the rest of the body. This is the second cycle. So this means that the blood is flowing from the heart through the heart twice in one complete circuit. Alright. So what are we going to write down in the answer? The answer to this question would be, this is going to be the answer. You can read it, that blood flows twice to the heart in one complete circuit of the body. First of all, it flows from heart to the lungs and then back to the heart. And secondly, it flows from heart to the rest of the body. Alright. Now, let's move on to the part B. Part B says that figure 1.2 shows the pressure of the blood flowing through the different blood vessels. What is it showing? It is showing the pressure of the blood flowing through the different blood vessels. As it travels around the body, venules are the narrow vessels that connect capillaries to the veins. So they have given you the information about the venules that are the narrow vessels which connect the capillaries to the veins. Alright, so this is the pressure graph. Let's read this graph first. That over here the blood pressure is given in kilopascals. Then we have the type of blood vessels over here on x-axis. So the highest blood pressure, that is till 16 kilopascals, is given in the arteries. Alright. And then the blood pressure starts to decrease. The blood pressure is around 10, 8 and 6 in the vessel labeled X. And then we have the constant decline of the blood pressure in the rest of the pathway from the capillaries to the veins. And we have the lowest blood pressure in the veins. Alright. So part 1 says blood vessels X in figure 1.2 supply blood to the skin surface of the capillaries and have a role in maintaining constant internal temperature. Thermoregulation. So we all know that the vessels that are responsible for supplying the blood to the skin surface and have their role in the thermoregulation are the arterioles. And what happens is that this is the artery. This is the artery. Then it gets divided into narrow arterioles. And then we have the blood capillaries. The network of the blood capillaries is like this. Great. We have the network of blood capillaries in the net form. And then the color of the blood starts to change from red to blue. The capillaries then join to form venules. And then we have the thick blood vessels that are the veins. Alright, pardon my drawing, but it is something of this sort. So the answer to this question would be arterioles. So let's write down arterioles over here. Now, part 2 says explain reasons for the changes in pressure seen in arteries in figure 1.2. So what are the changes in the pressure that are seen in arteries? First of all, we have to note that the changes in pressure is that the blood pressure in the artery is first increasing to the maximum, then decreasing, then increasing, then decreasing, then increasing, then decreasing. So this is actually what sort of the pattern? This is a pulsating pattern. Alright. So this means that the pulse is coming and then the pressure is increasing and subsequently the pressure is decreasing. Alright. So what causes such changes in pressure? Such changes in pressure are caused with the contractions of the elastic muscles present in the heart or the ventricles. Basically, when the heart pumps the blood, when ventricles contract and they pump the blood throughout the body, the pressure increases and there is a pulse. See this pulse. Alright, this means that the pressure is increasing and the heart has pumped the blood to the body. Then the pressure decreases. The pressure decreases when the heart or the muscle of the ventricle relax. Alright, the pressure and then pressure decrease and then see the pressure is subsequently decreasing. Why is this so? Because as we move away from the heart, the pressure in the vessels will decrease. Alright. So I hope this was clear. So what will be the answer to this question? Due to the contraction of the heart muscles. Pressure increases when ventricles contract and decreases when they relax. Right. Let's move on to the next part. Part 3 is using the information in figure 1.2 explain the structural adaptation of arteries and veins. So, what are the structural adaptations of arteries and veins? We have structural differences. First of all, let's discuss those. So basically, arteries have thick muscular walls, and via that, it is able to withstand the high blood pressure. All right. So next thing the arteries have is that arteries have elastic tissue that helps them to stretch and recoil, and they have a narrow lumen which maintains the high blood pressure. All right. So number one thing, they have thick muscular walls. Number two, they have elastic tissue to stretch and recoil, and number three, they have narrow lumen which helps to maintain the high blood pressure. And then let's talk about veins. What are the special adaptations of veins? The veins have large wide lumen to provide less resistance for the blood. All right. So what are the differences between the structure of arteries and veins? Basically, arteries is like this. This is the wall, and this is the lumen. All right. And vein is like this. It has a large wide lumen to provide less resistance. It has less muscular walls. It has more smooth muscle than them. And the next thing arteries have is that they have valves to prevent the backflow of the blood, which is the backflow of the blood is because the veins carry the blood away from the heart. Sorry, towards the heart, right? The veins are carrying blood towards the heart. That's against the gravity. So there are chances for the blood to flow backwards. So to prevent this thing, we have valves in the veins. So we will write the answer. So this is the answer to this question. You can read it from here. All right. Question number 2. Student investigated the digestion of starch. Figure 2.1 shows the apparatus she used. Alright, so the apparatus is test tube A and we have the test tube B. Alright, so the test tube A both the test tubes have the whisking tubing. This is the whisking tubing, and that is actually dialysis tubing. Alright, they are using the word dialysis tubing. Then we have distilled water outside, and we have the starch waved. We have the starch solution inside, and we have the distilled water outside. In the figure B, test tube B, we have starch plus amylase solution, and we have the distilled water outside in this tube as well. Dialysis tubing is used to represent the cell membrane. All right. Dialysis tubing material allows the small molecules to move across it, but not larger molecules. Test tubes A and B were set up as shown in figure 2.1 and placed in a water bath at 37 degrees for 30 minutes. Alright, the liquid out of the dialysis tubing in test tubes A and B was tested with Benedict solution at 0 minutes and 30 minutes. Means that the Benedict test was done before starting the investigation and then after ending of the investigation, after 30 minutes. Alright, so table 2.1 show the results. And we have test tube A and test tube B. So test tube A was blue at 0 minutes, and test tube B was also blue. Sorry, and at 30 minutes it was also blue. So what was present in the test tube A? Starch was present in test tube A, and Benedict solution gives results when there are reducing sugars present. Starch is not a reducing sugar. So it has no enzyme present in it, so it will not be digested into the simpler reducing sugars or the sugars. So there will be the results of the test will be negative. All right. So in test tube B, it was blue at the beginning and it turned red at the ending of the investigation. So using the information for the table 2.1, explain the reasons of the difference in the result for test tubes A and B. The test tubes A and B, we have discussed the reasons that Benedict's in solution A will remain blue and there will be no color change. Why is that? So because there is no maltose or there is no breakdown of starch. All right. Then in test tube B, the Benedict solution turned red after the digestion, which showed the presence of maltose. All right. This means that amylase has broken down the starch into maltose, and maltose is a small molecule, so maltose is able to pass with the dialysis tubing by what method? By diffusion. All right. Along the area, from the area of high concentration to the area of low concentration, or you can also use the word down the potential gradient. Alright, so we have to explain the reasons for the differences and the results. First of all, I told you the differences, what were the differences, and then we explained it. So the answer to this question will be that Benedict's in A remains blue as there is no maltose present, and Benedict's in B turned red due to the presence of maltose as the amylase broken down starch into maltose. Maltose is a small molecule, so it went out from the dialysis turbine for diffusion down the concentration weight. Alright, so you have to write this answer. Part B says complete table 2.2 by writing in the names of missing enzyme substituting products. This is a very easy part. Enzyme pepsin. So the pepsin breaks down proteins into the amino acids. Then we have fatty acid and glycerol. So what is the substrate for that? Lipids. Lipids are broken down into fatty acid and glycerol, and lipase is the enzyme. Alright. So lipase, lipids. Trypsin. What is trypsin? Trypsin is also a sort of a pepsin that breaks down proteins into amino acids. Then we have glucose. Glucose is a simple form of maltose. And what is the enzyme that is acting on them? Maltase converts maltose into the glucose. So, part C says, state the name of the structure that increases the surface area for absorption into the small intestine. So, small intestine has microvilli that increases the surface area for the absorption of the nutrient-rich blood. Right, question number 3, A. Figure 3.1 shows the part of classification with a diagram for plants. Complete figure 3.1. So plants are classified into flowering plants and non-flowering plants. Non-flowering plants are the ferns, and the flowering plants are normal plants. We label them as flowering plants. Name them as flowering plants. Now, flowering plants are further classified into monocotyledons and dicotyledons. Here we score 2 marks. Now, part 2 says describe 2 identifying features of the monocotyledon. Monocotyledon seed, I'm going to draw over here. It is like this. I hope that you have seen the seed of a corn. Everybody eats corns, right? So the corn is like this. If you have noticed that it contains a single leaf and it has the single area, like the structure of the monocots are not basically symmetrical. We cannot break the seed down into exact half. You cannot do that. Unlike dicotyledons. So the monocotyledons are basically one leaf plant. They have one seed leaf. Secondly, they have parallel veins. The veins are present throughout the structure, and they have adventitious roots. Always remember that they have adventitious roots, and they have vascular bundles throughout the stem as well. So we can write down so many of the features I have just told you regarding monocotyledon seeds, that they have one seed. Mono means one seed leaf. One seed leaf. What else? They have parallel veins. Some extra I'm writing over here, that they have adventitious roots, and they have narrow leaves, vascular bundles throughout the stem. Alright? Now, part B says figure 3.2 shows the life cycle of a fern. The life cycle of a fern has some similarities and some differences compared to the life cycle of other plants. The letters represent processes occur during life cycle. The haploid stage of the life cycle represented by N. The diploid stages of life cycle represented by 2. Very self-explanatory. Part 1 says the adult sporophyte has 1200 chromosomes in its body cells. Say the number of chromosomes in body cell of the gametophyte. So gametophyte is haploid, right? So haploid has half the number of chromosomes. So 1200 divided by 2 would be 600. So there will be 600 chromosomes in the gametophyte. Identify the process and define and describe the process in A. So A, we have 2 parts of this question. First, we have to identify and then have to describe. So what is happening in A? There is a formation of the gametes and then zygote and then adult sporophyte and then adult gametophyte. So what division is this? Meiosis. Alright. And meiosis is known as reduction division, in which chromosome number is half. This means that we have single set of chromosomes that is produced during the meiosis. That's why it's known as reduction division. Secondly, they produce genetically variant or genetically different cells. And what else? The cells that are produced are known as gametes, and they are spores in this case. The spores are dispersed by the winds, and then the further reproduction takes place. Alright. So we will write that. If we done this question. So answer to this question. Now part 3 says that state the name of the process. The zygote is formed. Zygote is a result of the fertilization of the 2 gametes. male gamete with the female gametes fuse or fertilize together and they form a zygote. So what is the name of the process named in D? It's going to be the fertilization. Part C says figure 3.3 shows the parts of two flowers from two different flowers of the same species. Alright. Describe what is meant by the term species. So we all know that species are a group of organisms that can reproduce or reproduce to give or produce a fertile offspring. So what is species? It is the group of organisms to produce a fertile offspring. This is a one-line definition which is going to give you 2 marks because it carries 2 points. Now part C2 says annotate figure 3.3 to show the mechanism of cross-pollination. Labeling the structures shown involved and draw an arrow to show the path of the pollen. So, cross-pollination occurs between anthers of one plant, that is this. This is the anthers, and then we have the ovaries and anthers, and we have the stigma. So the stigma is this. We are going to label this as S. Alright. So anthers and stigma should be correctly labeled and the line should be drawn from anther to the stigma. Alright. So this and this. So in this way we have the cross-pollination. Alright. Now draw X on figure 3.3 to show where fertilization occurs. We all know that fertilization occurs in the ovaries. Ovary is over here. So this is the X. Alright. Now, explain the disadvantages of cross-pollination compared with self-pollination. So in cross-pollination, what occurs is that basically the gametes or the pollens are dependent on the environmental condition or dependent on the wind to get transported to the desired mate. Alright. So for that, if the wind is not present, the fertilization cannot occur. So secondly, there is a lot of wastage of the pollens because there is less chance of pollination and cross-pollination. Because there are chances that they might end up into the wrong plant or wrong flower. So the pollen might get wasted. Secondly, the energy is required more in cross-pollination as compared to the self-pollination. Alright, and secondly, thirdly, it needs more than one plant. So we can write a lot of points in this question. Now, after this question, that will relies on when pollen might get wasted, more energy is required, and cross-pollination needs more than one plant. Now, question 4. Figure 4.1 is a photograph of a tree that has been girdled. Girdling involves removing a complete circle of bark and phloem from around the tree. So they are removing 2 things. Number 1 thing is that they are removing bark, and number 2nd thing they're removing is phloem. Alright? So what happens is, explain why the area above the girdle in figure 4.1 will become swollen. Why is the area becoming swollen? Use the term source and sink in your answer. So why is it becoming swollen? This is because what's happening, phloem is removed from the tree. Alright, so phloem is removed from here. There is no phloem over here, but see, there is phloem over here. So what will happen that what happens is the translocation takes place in which carbohydrates and sugars and glucose are made in the leaves through photosynthesis, and they are converted into sucrose for the transport, and the assimilates will be transported from the source, and roots will be the sink of the sink for the assimilates. What happens is phloem transport. Since phloem transports sucrose and amino acids, so they will transport, but everything will be collected above the girdle. All right. So we will write this as the answer. This is the 4-mark question. That we need to describe the translocation takes place in glucose made in the leaves through photosynthesis. We need to describe this, that when is a glucose made, it is made through the photosynthesis or in photosynthesis. It will be converted back to sucrose and transported from source that are leaves to sink that are roots. We need to specify what is the source and what is the sink. But all of the glucose or sucrose will get accumulated and above the girdle because there is no pathway. Alright. Now, part 2 says explain why the leaves of girdled trees are still able to receive mineral ions from the roots. Obviously, because xylem has not been removed. Only phloem has been removed. This is a very simple question. It's a very simple answer that xylem is still intact and mineral ions are transported by xylem along with water. So, So will be the answer to this 2-mark question. Now, part B says, explain why glucose mediated photosynthesis is required for absorption of mineral ions by the root. So what happens is that glucose that is made during photosynthesis is used during aerobic respiration to produce or to release energy. We're not going to use the word produce, we will use the word release energy. So ATP production takes place in which phase? In aerobic respiration that requires glucose that is supplied through photosynthesis, alright? And then this ATP is important, or this ATP is required for the active transport of the mineral ions from low to high concentration radium. So we need to write all of these things over here, all of the details over here, that this will be the answer to this question, that glucose is required for aerobic respiration to release energy, and ATP is used for active transport of the sugar ions against the concentration gradient. Alright. Now, state balanced symbol equation for photosynthesis. So, students, you need to learn this equation by heart, that 6 molecules of carbon dioxide react with 6 molecules of water to give 1 molecule of glucose and 6 molecules of oxygen. So, this is a very much crammed and oldie sort of an equation that we need to learn, that 6 carbon dioxide plus 6 water gives rise to 1 molecule of glucose plus 6 molecules of oxygen. Alright, this is the equation. Now, question 5 says that the eye is a sense organ that's a part of the nervous system. Explain why eye can be described as a sense organ. So, sense organs are those organs which carry the receptor cells. So, eyes also carry the group of receptor cells that detect or that respond to the light, that is a specific stimuli, right? So, we will write down that eyes contain group of receptor cells that detect and respond to the light. Part B says take 2 parts of the central nervous system. So CNS comprises of 2 parts, that is brain and the spinal cord. Alright. Part C says figure 5.1 is a diagram of a section through a human eye, and draw an X on figure 5.1 to identify the blind spot. Blind spot that is on the scopia is present over here. So this is X. Alright, now they're saying table 5.1 shows parts labeled in 5.1, they are named in their functions. Complete table 5.1. So H, what is H? H is retina. Over here, the innermost layer. Wait, let me make it more prominent for you, that this is the innermost layer, and the innermost layer is known as retina. Alright, so this is the retina. It contains rods and cones. That's it. Then transfers impulses to the central nervous system. So the central nervous system transmission of impulses takes place through the optic nerve, and optic nerve over here is the G. Alright, this is the optic nerve. So the letter will be G, and this is going to be the optic nerve. Then we have the cornea. So cornea is, first of all, we have the retina, and then the cornea is present on the anterior part of the eye, and it is present over here above the, like after the lens, we have the cornea, and that is the D. Alright. So cornea is the D. So what is the function of the cornea? It refracts the light. Basically what happens is cornea helps to or allows the light to enter the eye. So it has the function of refraction. So refracts the light. That's it. Part D. State the name of the 2 effectors that contract and relax during the pupil reflex. Effector muscles are present in the eye that are known as circular muscles and radial muscles that contract and relax during the light reflex or the pupil reflex. So circular and the radial muscles. Alright. Now, part E. Explain why a person is unable to focus on distant objects and suspensory ligaments become permanently overstretched. What are the suspensory ligaments, students? Suspensory ligaments are present over here, and these are the suspensory ligaments. So what happens is, what's the question? Question says that if the suspensory ligaments are overstretched, what will happen? They will not be able to focus on the distant object. So why is that? So we need to explain that. Alright? First of all, we need to understand the normal phenomena which occurs when the eye has to focus on the distant objects. What happens is that the ciliary muscles contract. Alright? The ciliary muscles relax and suspensory ligaments become taut. And when they become taut or tight, the lens is stretched. Alright? And the angle of refraction is changed in a way that it focuses on the distant objects. So when the suspensory ligaments can no longer or they are overstretched, due to which they can no longer become taut, so ciliary muscles will relax, so since the suspensory ligaments will no longer become taut. Hence, what will happen? Lens will not be stretched and it will remain widened. Alright, and angle of refraction will also not change, and the angle of refraction will remain unchanged. Alright, so the answer to this question will be that ciliary muscles relax and suspensory ligaments can no longer become taut and lens remains widened. Angle of refraction remains the same, but there is no accommodation for distant objects. Alright. Now, question number 6. Rice. Golden rice is a type of rice that has been genetically modified containing a nutrient called beta-carotene. Figure 6.1 shows the genetic modification process used to produce golden rice. So what is happening? This is a bacterial cell. The plasmid has been taken out. The plasmid is then cut using restriction and manipulated enzymes. And then the plasmid has got the foreign gene or the plant DNA, more specifically, administered in itself. And then they are mixed together with the material. They're entered and they're taken back of the bacterial cell. And then rice plants infected with genetically modified bacteria are breeded in the laboratory. And then what happens is the rice plant with the ability to produce beta carotene are grown, and they are grown in a multiple amounts. Alright. This was about figure 6.1. Using information in figure 6.1 complete sentences to describe how rice is genetically modified to contain beta carotene. We have just discussed this thing above that part labeled A, bacterial cell is a, what is A? It is a plasmid. Alright. So this was very easy. It is a plasmid. Then part A is extracted and cut using what enzyme is used? I just named it that the enzyme is known as restriction endonuclease. Alright, enzyme forming sticky ends. Sticky ends are formed so that it may be the plasmid may incorporate the foreign gene or the gene from another organism into itself. Alright, now, so part A is extracted and cut using enzyme restriction endonuclease forming sticky ends. Part labeled B is a dash that holds for products of beta carotene. So beta carotene is a protein. So what will be the coding thing for the protein? Gene. Alright, so gene that goes with the production of beta carotene. Enzyme used to cut part A is also used to cut part B for the plant DNA. Part B is inserted into the plant part labeled A using enzyme. Ligase is used to glue together two things. So look back over here. That this thing is sticked or glued with the cut ends of the plasmid. So we always use DNA ligase for gluing two things together. Alright. Now moving ahead, this forms a part labeled C, which is a recombinant plasmid which has the gene from other sources known as recombinant plasmid. Part C is put into a bacterium. The bacterium is taken up by rice plants, giving them ability to produce beta carotene. Tissue culture is in the form of dash reproduction that is used to produce many identical plants producing beta carotene. So the very major clue for you to identify whether they're talking about asexual or sexual reproduction is that they're saying that many identical rice plants, identical species are produced in the asexual reproduction. Alright. These are the blanks I have filled. Alright, so you can read it from here. Now, part B says, apart from structural features, state two reasons why bacteria are useful for genetic modification. So the bacteria are useful for genetic modification because they're able to reproduce rapidly because they have binary fission or they reproduce asexually, which is a rapid form of reproduction. Secondly, there are no ethical concerns. If you will use humans, so obviously this will give rise to many ethical concerns. Thirdly, they have the ability to make complex molecules and they don't require too much space, right? So what are the advantages? The advantages are that they reproduce rapidly, and there are no and some extra points I'm writing for you that they have ability complex point. Now, part C says, discuss the advantages of the genetically modifying rice plants to produce beta carotene. So, the genetically modified the disadvantages they are talking about. Part C says that discuss the disadvantages of genetically modified rice plants to produce beta carotene. What are the disadvantages? Disadvantages for the genetically modified crops or for the genetically modified rice plants are that additional energy content that is added is might be too small for the people to use, right? And the seeds are very expensive to buy. So for farmers, it's not very cost effective. So this is going to cost them a lot of money. Farmers are unable to save the seeds. Alright? Secondly, that there might be side effects that are unknown or that are the users or the consumers will be naive about the side effects for the genetically modified species. Secondly, what happens is that when we use genetically modified crops or when we generate genetically modified plants, they cause a reduction in genetic diversity, or in other words, they cause the reduction in the biodiversity, right? So these were some of the reasons. Along with this, there are also ethical concerns. Regardless if we are using if you're using the bacteria or the humans, regardless of the fact, people have the ethical concerns for buying the genetically modified products. All right, so you can read the answer from here that carries 3 marks. Now, part D says beta carotene is required to produce vitamin A. It is essential for eye function. It is a nutrient required to prevent scurvy and rickets. So scurvy is a disease in which there is a bleeding from gums, and bleeding from gums is prevented by using vitamin C. And rickets is a disease in which there is a defective mineralization of the bones in the kids because of the deficiency of vitamin D, which results in the inabsorption for the calcium in the body and results in the defective mineralization of the bones. Alright, so this marks the ending of our paper, and this paper carried 6 questions, and I hope that I hope that you understood each and every part of this paper. And if you guys have any questions, you can always ask on the comment section below, or you can also email me, or you can also contact me. I will provide you my contact details over here. So you can always email me over here if you have any queries regarding the paper 4 or paper 2 for the biology subject. And if you have any queries, you can always comment in the comment section below, and we will try to address all of your queries. So this was it from my side for today. We will meet in the next video with another paper. Till then, goodbye and good luck for your exams and for your preparation.